Thank you everyone for joining tonight. My name is Ben Wade. I'm an applied sciences engineer at Crux Product Design. And tonight I have the pleasure of, for the next 45 minutes or so, giving you a crash course in inhaled drug delivery and how we can use sort of cutting edge simulation tools to accelerate the device development side of these really advanced drug delivery pipelines to sort of improve the patient's condition and make better and more efficient devices. So I thought it'd be quite a good idea to give a little brief introduction to Crux, as not all of you will have heard of Crux before. So Crux are a consultancy based in Bristol. So myself and Ollie work there, and we're about 100 strong now. And we are designers, engineers, researchers, and usability and software and electronics all under one roof. So we can take something from a blank sheet of paper all the way to commercialization of a mass manufactured product, and we can get involved at any stage in that process. And we've been trusted for over 20 years by some of the world's largest pharmaceutical companies and fast-moving consumer goods companies. And our claim to fame is Crux designed the 2012 Team GB cycling helmet. So that's why that logo is on there in the bottom left. Just to showcase some of the sort of the fun we have on the applied sciences team, this is some of the work that my colleague Joel has been doing a lot of work on. So looking at subcutaneous injections into animals for delivery of slow release drugs. So this is an ex vivo CT scan image of a subcutaneous injection and an equivalent FE model where we can predict the bolus shape and back pressure of the injection, which are quite important parameters when delivering these sort of advanced therapeutic drugs. Our software, electronics, and usability team also make really cool stuff. So this is our Crux IoT device that we coin, which is a small embedded software with connectivity, so we can get live IMU data and load cell data. And we primarily use these in human factors studies to quantitatively assess how users interact with devices so that we can design the devices better. In the bottom right, this is sort of our bread and butter. So this is our explicit dynamic FEA models of auto-injector assemblies, typically around 20 components. So this particular example is showing a drop test where we've dropped a insulin pen and we're assessing if the device will fire by accident when it has been dropped, because that would count as a failure. So we can assess all of these things in silico with some in-house material characterization that we do at Crux. Moving on to more kind of the fluid mechanics side, which is kind of the focus this evening. This is a nice example of how we do sort of engineering communication. So we take something which is computational cells and colors, and we can transform it into something which looks realistic. So for non-technical stakeholders, we can really get their buy-in and really get the team the appreciation that they deserve, because it becomes more tangible and they get more recognized and more funding. So this particular example is looking at nanoliter scale glue deposition on a neurovascular guide wire. So this would be immeasurable in real life and not very repeatable. So we can use CFD models to predict the volume of the deposition, but also the wicking behavior due to the surface tension. Looking more at the process side, this is a dry bead milling operation. So we're going to be talking about dry powder inhalers later in the evening. So this is one of the processes by which we make dry powders, so very fine micron size particles. So we can model these scenarios, optimize machine parameters, and also just design better machines. And then one final example, something I've been doing a lot of recently, is injectable suspension modeling. So looking at the rheology of injectable suspensions and looking at the injection events such as cartridge piercing and needle occlusion, and using these in silico methods to risk screen the formulation and device combinations before we get down to clinical trials. And just provide some context as to why I'm kind of showing you that and why we think these things are so important and why Crux has a unique space in the market. Is because simulation has the potential to reduce or eliminate the need for physical prototyping and testing, and to rapidly and cost effectively evaluate more design and clinical use variations than are feasible using traditional methods. So in short, using simulation tools, we can go faster than we could in real life, and we can get more data and measure things that we can't measure in real life. And I highlight the word rapidly there because these tools are used ubiquitously in other industries such as aerospace and automotive, and we think they're chronically underutilized in the med space. So that's something that we bring to the table at Crux, and that sets us apart from our competitors. And then something to keep in mind, the topic of today's presentation. So we're going to be focusing on metered dose inhalers, or puffers as they're commonly known. So the carbon footprint from one typical PMDI, which a user can go through in a week to 2 weeks, is the equivalent of driving a car for around 180 miles. And this is because of the propellants that are used in these inhalers. So we moved away from CFC propellants in the 80s because of the Montreal Agreement. We're now on HFA propellants, but they still have a significant global warming effect. So we're using these tools to develop the next generation of devices with net zero propellants. And that's kind of the focus of today's talk. But actually, the majority of the content, which I'm going to begin with, is actually providing context as to why we need inhalers and then how you can get drug to the lung. And then that will be followed up by an explanation of the model showcase, which we've set up, and then some results exploration to finish, and some conclusions on why we think these tools are so important. So kicking off with the sort of background and literature review, a very sensible place to start is looking at the actual human anatomy. So what is the airway? So we have the nasal and oral cavity in our heads, which join at the back of the mouth, and these travel down your trachea. And from this point onwards, the airway splits. So mathematically speaking, it bifurcates, and it does so 24 times. And in doing this, it significantly exponentially increases the surface area of the lung, and it slows the air all the way down so we can get gas exchange in these little sacs, the alveoli right at the end of the lung. And there's some immense statistics about the lung. So supposedly the length of all the airways, if you chain them all together, is around 2,400 kilometers, which I couldn't quite believe when I first read it, and I'd like to know how they measured that. And then secondly, the surface area. So if we took the surface area of the lung for an adult, that's estimated to be roughly the size of a tennis court. So really the big surface area and the big area which we need to get drug to is deep down these branches. Imagine a tree which just keeps splitting and splitting and splitting. We want to get all the way to the end where the leaves are, from the roots, from where the trunk leaves the ground. Looking at this through the lens of fluid mechanics and talking about the flow regimes which we'd expect. So we can get turbulent flow predominantly in the nasal cavity and the oral cavity. And then the deeper in the lung we go, it becomes deeply laminar quite fast. So in order to model this in one go, you would need to model the turbulence, the transitional flow, and also the laminar flow as you sort of descend deeper into this geometry. For those of you that have performed CFD, you know that's a big challenge, and the sheer scale of and complexity of a model like this is sort of unattainable in one go. And kind of, why do we need inhalers? Well, predominantly, the majority of inhaler sales are to treat a condition called asthma, which I assume most of you have heard of. But this comes under an umbrella of conditions called COPD, so chronic obstructive pulmonary disease. In the UK, this affects around 1.2 million people, and it's basically an inflammatory response for the most part. So it's your body trying to protect itself from something that it's encountered in the air or in your sort of a systemic response, and it kind of just closes up. And in closing up these airways, you're increasing the resistance of that airflow, and you're making it a lot harder for the patient, for the user to breathe. And during particularly chronic episodes, it can be very, very difficult to breathe, and can lead to more severe conditions. And actually, this is the second most common cause of emergency hospital admissions in the UK, accounting for around 1 in 8. And there's an increase in prevalence of COPD of 40% by 2030, which costs the NHS a significant amount of money. So we need to make better devices which can deliver these drugs to where they need to be, so that we can kind of assess and mitigate these costs and clear up the NHS. And as it sort of alludes to in that comment, these numbers are only going one way, for a variety of reasons: destruction of the gut microbiome, sort of increased industrial chemicals everywhere, foods, the air, water, and something I've been reading about more recently was actually sort of cumulative generational trauma. So there's just the buildup of stress and your sort of decreased bandwidth to deal with that stress. But that's a subject of a different talk, which I'm not professionally qualified to give. So we need to deliver drug to the locally acting site to reduce the inflammation, a bit like how you put a cream on a rash. So if we just took oral medication, so if we swallowed a tablet and digested that tablet, it would affect our whole body, and that's not what we want. We only want to treat the lung. So we need to get drug directly to the lung, which means inhaling it. And how we do this is through devices called inhalers, which produce aerosols. So all an aerosol is a liquid droplets or solid particles suspended in a gas. So a spray or a sort of a dry powder aerosol. And these are the three kind of main types, which we'll focus on the top one today, and then the bottom two will also get a mention. So how to actually get drug down there? And this is something that always kind of surprises people, is just how difficult it is. So I've already spoken about the kind of geometrical challenges in terms of the bifurcations and the branches and the laminar flow. And this graph does a good job of kind of illustrating the challenges. So on the y-axis, we have the percentage of deposition, and on the x-axis we have the diameter of the particles, and the scale here is in microns. So you take a meter, you divide it by 1,000 to get 1 millimeter, and then you take that 1 millimeter, divide it into 1,000 parts again to get 1 micron. So they're the kind of scales that we're speaking about. So this black line is the total deposition. So this is stuff that will stay in the body, it won't come back out. And we see this kind of peaks around 4 to 8 microns. And then the lines below this show where the drugs land within the respiratory system. So if we look at the top of the airway, the mouth and throat, we have this big right angle bend where the mouth and the nasal cavity change direction to go down your body. And we see a huge increase in deposition for larger particles. And this is due to inertial impaction. So you have a big particle entering the mouth and throat. It's moving very fast, and it literally just can't change direction at the back of the mouth. So you get just a big splat in the back of your mouth. And the nasal cavity, which we'll come on to in a couple of slides, its job is to filter out particles that you shouldn't be inhaling. So it makes a very tricky, convoluted path for particles to reach the deep lung. Next, if we look at the first few branches of the lung, so where it starts splitting, but not down to the really big surface area, we see peaks around 3 to 5 microns. And finally, if we look at the pulmonary region, so right at the end of our tree, where we want to be delivering the drug, where all the surface area is, we see sort of between 1 and 3 microns. And actually, what I'd like to point out, and is one of the non-obvious things here, is there's a complete drop-off in deposition at the sub-micron scale. And what happens to these particles? Well, they just get breathed back out, so they just don't land. They're happy just staying in the air, floating, they just come straight back out. So as engineers, we want to deliver drug here, and we have a really small window to do so. So we need particles between about 1 and 5 microns is what we'd aim for in order to get drugs down to the lung. And from a technical device standpoint, transport, usability, that's very, very difficult. And not only is that the only requirement, it matters how fast these particles are going. So what I've shown on the right here is a simulation of a dry powder inhaler. And this geometry you see here, we'll get familiar with a bit later. This is a CT scan of a human airway. So this bit here is the mouth. This bumpy bit here is your tongue. And then this is your trachea and your larynx here, which goes down to your airway. And there's an overview of this geometry in the top right. And what this circular video here is what we call the swirl chamber in the dry powder inhaler. So powder is going to get picked up from these channels and it's going to smash against all these walls and hopefully get broken up to between 1 and 5 microns so it can make its way around this bend and down to the lung. But the velocity that the particles leave the inhaler is very significant. So this concept of inertia, so the resistance to change in direction would be a function of the diameter of the particle and the velocity. So on the x-axis we have the velocity or the volume flow rate, and the y-axis we have the particle diameter. So big particles moving very slowly aren't deposited, but then smaller particles moving very fast are deposited. So ideally, we want small particles between 1 and 5 microns moving very slowly. But this poses a ginormous trade-off, which, if you'll pardon the pun, is the crux of the problem. So we want a lot of flow energy to make a good aerosol between 1 and 5 microns, but we don't want a lot of flow energy going into the patient, because it will be too high velocity and all this drug will just leave and it'll not change direction. It'll just sort of splat at the back of the mouth and it will have no therapeutic effect. It will not improve the patient's condition. And it could even have adverse effects, such as giving the patient oral thrush, which isn't very pleasant. So how do we kind of test inhalers today? These experimental devices and drugs, it's very unethical just to test them immediately in humans. So the industry standard is something called a cascade impactor, which is what this image is here. And what this pipe at the top is if I just go back to this slide, it looks kind of similar to this shape. So this is kind of meant to represent the mouth and the trachea. And then this system down here is just a series of cups with increasingly smaller holes, and this is designed to filter out and categorize particle sizes. So if particles make it past the upper airway and into this impactor, we can get a read on how big the particles are, and from that we can say how well the device performs and how much of that drug we'd expect to reach the deep lung. But that's about it. And even this induction port here is pretty advanced. Before this, they were just using a right angle steel pipe, which looked like something from the 40s, and had no resemblance to anatomy whatsoever. And increasingly now, or actually not even increasingly, I've only seen one outfit do this. We're using these split throat models. So if we want to know how much drug lands in the mouth, in the back of the mouth, in the trachea, we have to make one of these split throat models, machine it from metal, covered in silicon gel to represent the mucosal lining, and then actuate an inhaler, take it apart, do a chemical assay on each part of that, and then get a result. And that's one really intensive test, which also has loads of experimental errors. So in summary, it's really hard to test inhalers, and it's something that we're not very good at. And if we move on to kind of the next level, which would be once we know the device and the drug is safe, where we can test it in humans, kind of the gold standard of this, up until very recently, is something that we refer to as in vivo radio-labeled scintigraphic images. So the radio-labeled bit just means that we tag the drug with a radio-labeled substance. We get a user study and a patient to inhale, and then we take a scintigraphic image, a bit like an X-ray, and where the drug lands, it kind of glows. And this is a 2D image, and they've outlined the lungs here as well. So these are from 3 different types of inhalers, and the brighter parts are the better drug delivery. And just to point out some common features, so we see a lot of deposition in the mouth. So this is the parts in sort of the top right of these images. but also we see a lot just below the lungs in the stomach. So this is drug that's landed in the upper airways and is just being swallowed and will be useless. It will not improve the patient's condition. So we see relatively poor performance for the dry powders and the PMDIs. This device here is called a soft mist. And just as I said before, this ticks the box of making a really good aerosol that moves slowly. So at this point, I'd love to open up to the floor, and any guesses for the label claim of an inhaler, which actually reaches the lung. So say if your inhaler says 100 micrograms, how much of that 100 micrograms actually reaches your lung in a typical inhalation? Any takers from the audience? No? Quiet one tonight. Ollie, do you want to have a guess? You might know the answer.
Let's give it a solid 25%.
20. Cheers, Alex. Cool. Yeah, so inhalers are pretty bad. And a lot of people don't realize this. So we get a lot left in the device, we get a lot in the mouth and throat, and we don't get a lot in the lungs. Even for this really good soft mist inhaler, we're looking around 50%. But for normal devices, which the NHS prescribe, we're looking at between sort of 10 and 30%, 40% if the stars align and you do everything right, which no one ever does. So you need to do your breath holds, you need to do your shaking, your drug prep, you need to follow all the steps to the T, and then you'll get a good dose. But this is really poor. So in terms of label claim, we're putting more drug in than we need to, and these devices aren't very robust between users. So this is kind of where simulations come in. So we've spoken about how hard it is to test these things in real life. We've spoken about how expensive and difficult and possibly unethical it is to test these in humans. So if we can reproduce all of these results in computers using physics-based simulation software, we can accelerate this drug development. And there's three main areas for inhaled therapeutics where we can focus these efforts. We have the formulation, so the powder or the liquid. We have the device, so we can tune the device resistance, the way that powder is broken up within the device, any leak paths. And finally, we can predict the deposition, which is kind of the topic of this presentation this evening. And these have been around in the literature for some time now, and they're becoming quite mature, but they haven't made their way to industry. We have a nice example here of a unsteady meter dose inhaler model, which looks very pretty when it's actuated. It had a 280-hour wall clock time because of the relatively high fidelity LES turbulence model. And we've actually had in vivo validated studies against those radio-labeled images. So these models, these techniques are capable of giving us the results that we need. But to date, they've only been used in anger at devices which are already on market. So they're only now being used to create better devices for the future. And a quick aside, so we've been talking about lung delivery, but this alien-looking thing on the screen is actually your nasal cavity. So this is reproduced from a CT scan of a real person. If you make a fist, just for scale, it's roughly just smaller than your fist, and it sits right behind your nose, behind your eyes. And its job is to rapidly humidify the air and filter out any particles. That's why there's all these weird shapes. So when you have a runny nose, all of these surfaces will be producing mucus to try and get rid of those particles. So you keep blowing your nose, you keep sneezing. It's just trying to purge the things that shouldn't be inside the body. And of particular interest is these parts right at the top. And these are called or is referred to as the olfactory region, which is one of the few regions in the body and the only region in the airway where we can bypass the blood-brain barrier, so we can deliver drugs directly to the brain. And this is becoming increasingly important for the delivery of neurodegenerative drugs for treatments of conditions like Alzheimer's. But as you can see from the shapes of these geometries, it's very hard to get drugs past all these shelves all the way up to this top bit. But we can use models to try and improve these devices. And I wanted to touch on, to finish off this literature review, to hit home the power of these models and kind of set the stage for why they're so important. So this was a landmark paper from 2016 looking at one of the old MDI inhalers, so CFC propellants. And this particular lab managed to model the whole lung. So they resolved in 3D CFD down to about the 8th generation. And from then on, they used a 1D lung model, so a bit of a sort of stochastic lung model to predict particle deposition based on what left the CFD model. And they were managed to pull out for each generation in the airway, so generation 1 through 7 and 8 through 15, how much drug landed there. And it's illustrated in these images here. So in the mouth throat, the black is drug. So we see a very high concentration of drug where it's not needed. And the deeper in the lung we get, as the surface area increases and the amount of drug that's getting there decreases, we see a massive fall off in delivered concentration. And there's no clinical evidence that drug delivered in these concentrations has a clinical benefit. So this paper really, called the bluff and pointed out that the inhalers aren't delivering enough drug to where they need to deliver. I think this is a really great paper, definitely worth checking out if you're interested in this area. So now I'm going to move on to the case study. So we developed this as kind of a credential at Crux, and it kind of talks through the model setup of a model like this, where we can predict deposition in anatomical geometry, and I'm going to talk through some of the physics and some of the results. So how does a PMDI work? Well, you have this metal canister, which you have a drug formulation or a suspension, and this is held at elevated pressure, so between 3 and 5 bar typically. And the propellant, when it's held at high pressures, is very happy being liquid. So you can shake it, you can slosh it, it's happy, it's very content. But as soon as it leaves the metering valve and comes into atmosphere at atmospheric pressure and temperature, it wants to be a gas. So it literally just explodes. And then the inert drug stays as a liquid. So the explosion of the propellant propels the drug into the mouth. So you get a nice fine aerosol at the click of a button without having to breathe, which is great. The downside is, as we'll come on to, is that jet, that aerosol, is very, very fast. So it can leave up to 80 meters per second, the particles. So very fast, and why I should use a spacer, but I'll come on to that a bit later. So how do we model this in CFD? Well, if you know computational fluid dynamics, you'll know that spray modeling is very difficult, especially when there's phase change. So we need to characterize a spray and use this as a model input. So we're modeling the droplets using a Lagrangian phase of liquid droplets, and we have phase change enabled. So these droplets can evaporate and coalesce as they move through the domain. So the propellant, which is HFA vapor, enters the domain at its boiling point around 250 Kelvin and 60 meters per second. And as it leaves and travels through the domain, these particles can evaporate. But the ethanol, where the drug is suspended or dissolved, does not evaporate. So this is illustrated by the orange disappearing, but the black not disappearing. And this is accompanied by some HFA vapor inlet, which is a continuous phase. And this is just modeled as a background spray, as not all of the flow leaving the nozzle will have drug suspended in it. So the model inputs, as I just said previously, we need to characterize a spray. So this is work done out of Loughborough, where I graduated. They characterized MDI sprays using phase Doppler anemometry, so some quite advanced laser techniques. where you need to take several measurements at different distances from the orifice, and you can't get too near the orifice because the velocities are too high to pick up a good signal. But we can get things like liquid mass flow rate, cone angles, and droplet velocities. And if we accompany this with an anatomical airway, which has been reconstructed from a CT scan of a real person, so we're going down to, I think, this 6th generation here. And just to point out, so we have 2 lungs, a left lung and a right lung. Our left side has what's referred to as 2 lobes. So it has this branch here, which splits into 2 branches. And this is the left upper and left lower lobe. And the right side actually has 3. And the reason that your left side has 2, it's got a little space for your heart. So there's kind of this asymmetry between these 2 halves of the lung. Boundary conditions. So to save on compute, we're modeling this as steady state. So we're modeling near peak actuation. So a 15-degree nozzle angle at a velocity of 60 meters per second. And we're modeling the aerosol with 5,000 Lagrangian droplets with a log normal PSD, mean particle size around 5 microns. And as I just mentioned previously, there's an asymmetry in the lung. So each bit of the lung will pull a different amount of air. We can actually account for this using the mass flow outlet boundary conditions based on these percentages. So this is the percentage of volumetric flow rate which these parts of the lung, so each lobe, will pull. And we're just pulling air in from ambient atmosphere at the top of the inhaler canister. And one thing we're not modeling here is atmospheric humidity. So we're only modeling the evaporation and condensation of the propellant. So just some solver methodology for those that are interested. So we have what we refer to as 2 phases. So we have the continuous phase, which is the finite volume gases within the model. And then we have the discrete phase, which is our Lagrangian droplets. So these move throughout the finite volume model. So from a solver methodology, this steady state approach, we're going to solve the boundary conditions, so without any droplets. We're then going to ask the solver where the particles would go in that flow. And then because we're using 2-way coupling, we're going to add the effects of those particles back to the continuous phase. So these are added back as source terms, and that in turn is going to change the solution of what's going to happen to these gases. So we iteratively solve this until we reach convergence for both phases. So until the prediction of where the particles land doesn't change. And that's kind of the intent and our strategy for solving this efficiently. Otherwise, we'd be waiting a very long time to get these results. So if we look at kind of what's going on in that continuous phase, so the gases, this is a side view of the upper airway. So we have the tongue here, and then this is the oral cavity, and this is the nozzle of the MDI device. So you see extremely high gas velocities at peak actuation moving directly into the tongue. This was just assumed as horizontal. And this kind of directly correlates with the temperature and concentrations of propellant, so the mass fractions of HFA 134A. So we see ambient air come in with no propellant, and we see this rapid mixing in the oral cavity and some recirculation in the upper mouth area, and then some flow separation behind the tongue, where we would expect to see a lot of impaction towards the back of the mouth. And one of the artifacts of modeling this is steady state, so not as a transient plume, is that we get a lower than ambient downstream temperature because we're constantly jetting in propellant at 250 Kelvin. So the product of these two will reach a lower than ambient temperature downstream, but that's something that we take into account when we're interpreting the deposition results. We do deem this to be sufficient to get a read on the ratios of deposition, so where these particles will go. Moving to the discrete phase, so these liquid droplets, what happens to them? Well, as you can see from this image, and as I was saying right at the start of the presentation, they don't change direction. So I've colored this with respect to how much propellant there is. So red particles have a lot of propellant left, which means they're still big. They're still a big diameter. They have a lot of mass. And I've made the smaller particles, or the ones which do evaporate, more transparent. So we see a huge jet of very large particles which don't evaporate, because this area of the flow is low temperature and high concentration of propellant. And one of the great things about simulation is, for every one of these particles, we can plot as a distance from the nozzle the droplet diameter and the temperature of that droplet. So we can verify that our evaporation model is correct, and we can look at what point these particles get small. And using insights like this, we can start to recommend instructions for use, but also use these as knowledge for designing the next generation of inhalers. So the important bit, and what we alluded to earlier, is this regional deposition. So the circle in the middle of the screen is the tongue. So just as we were mentioning, there's a big splat on the tongue of these particles which don't change direction. And if we look at the bigger picture, again, we see a lot of upper respiratory tract deposition, just as we saw in those gamma studies, and some deposition mainly at the bifurcations, so where the airways split. And because this is a numerical model, we can get quantitative data on every one of these particles. And this is something that you just can't do in any sort of clinical study. We can look at exactly where all of these particles landed and quantify them. So we see huge deposition for a range of injection angles. So we're trying to aim this up over the tongue. And we do see a decrease in oral deposition, but then an increase again where we're actually firing into the roof. And not only can we get upper respiratory tract deposition, but also what we refer to as lobe penetration rates. So for each one of those 5 lobes, we can predict how much drug gets to that specific lobe based on our inhalation and based on our spray. So ideally, we'd want the proportion of drug going to that lobe to be matching the proportion of air that it's receiving, and the proportion of surface area which that lobe has. And these are all things which we can get from the model. And really the takeaway here is just how sensitive this system is to injection angle. So 5 and 10 degrees, we get pretty good lobe penetration. So the drug's going where we want it to go. But then anywhere outside of this, we're hitting the tongue or we're hitting the roof of the mouth, which isn't what we want to do. So obviously this is without a spacer. So if you use a MDI inhaler, use a spacer, it gives the propellant time to evaporate to get a nice, fine, slow-moving aerosol, which is going to get to your lung. But, like I said before, we're really using this to advance the next generation of all inhalation devices, because we can get insights like this, which we just can't get in real life. So just to throw up some conclusions, recognize we're getting towards time now. So really this study has highlighted the user-dependent sensitivity. So the user interaction, and something as small and sort of nonchalant as how you hold the device can have really significant effects to the therapeutic efficacy. We can digitize anatomy. So simulations come a long way from just modeling sort of square ducts. We can model really complex geometries, split them up into good quality finite volume meshes, and model the sufficient flow physics. And as I've been banging on about today, we can use tools like this across a range of applications to de-risk next-gen inhalation technology. And this is an example we made of nose-to-lung delivery, and this was with the Bill and Melinda Gates Foundation. So looking at delivering broad-spectrum antiviral drugs to the whole airway. And we can actually tune where the deposition will occur based on the particle size distribution. So if COVID-25 comes along, we can produce a dry powder with the sufficient properties to get drug where it needs to go, be that prophylactic or antiviral capacity. Amazing. Hopefully no one's asleep. Thank you very much for listening. More than happy to take any questions, or feel free to put them in the chat, or reach out to me personally on this email or on LinkedIn. Yeah, thank you very much for listening.
Thanks, Ben. That was incredible. It's amazing the sorts of insights that we can get from this type of analysis. Are there any questions that people would like to ask now? You can just unmute and ask them, or drop them in the chat. Read this one out from Alex Wood. We've got how long have you spent on this, and how much work remains? That's a bottomless question, I'm imagining.
Yeah, glass half full kind of guy. Yeah, we've come a long way. Yeah, I think the literature's come a long way in the last sort of 10 years, moving from sort of these two equation turbulence models to looking at actually eddy resolving models like LES, so we're improving the fidelity and the physics. So the next step now, so now that we can predict where the drug will go, is predicting the response of the body. So predicting the amount of drug in the blood based on where the drug lands. So that's kind of like the next frontier. And there's been mixed results so far, but that's something that the industry is very keen on doing because it'll save them their money. So your answer of how much work remains in terms of duration, tough to say, but I don't think it'll stop anytime soon, which is quite exciting. Joe Bornoff, speaker 2.
Hi Ben, can you hear me?
Yeah, I can hear you.
Okay. First of all, great presentation, great work. I always love a good CFD model, so it's great to see. I got a couple of questions. Well, it seems like the tongue is the main thing that stops the drug from getting further into the airways. I know you looked at this in a sort of steady state environment, but do you believe that your model could be extended to look at kind of how to inhale, how to best inhale the drug to improve that the reaching of the drug further into the airways? Sort of wall motion, if you could like move the tongue somehow transiently to change how the drug is moved from the mouth down into the airways. Is that something that could be done, could be looked into?
Yeah, I think it could be done. I think the question then becomes, how do you get someone to do that when they inhale? But yeah, it's definitely something that could be done. So we have, with our clients, developed unsteady models which resolve sort of temporal plume development, and you see kind of an upwards washing against the tongue as the aerosol plume develops and moves down, kind of as like a big slug. So there are things we can do if we have the model inputs. I think it's just a factor of, you know, how much compute do you have, as you'll know.
Yeah, exactly. Yeah, it's a fair point. And I guess you're getting, as you say, enough information from your steady state simulations that it might not be worth it in the simulation world. Thank you very much indeed. Yeah, as I say, great presentation. Good work.
No worries. Cheers, Joe.
Got another question from Alex, which is, if you're interested in getting the drugs to the blood, then why are we inhaling rather than just injecting them?
Great question. Again, not one that I think I'm qualified for. So good question. So we want to deliver drugs to the lung, so to the lung tissue, and looking at blood plasma levels, so looking at pharmacokinetic studies, we can tell how fast that drug is being absorbed into the circulatory system and whether it's saved. So we're using the drug levels in the blood as the body's trying to clear and process these drugs as a proxy for how much drug got there. So it's kind of like, I don't know, looking at how sweaty your t-shirt is after the gym as a proxy to like how hard you worked. It's just a way of determining the impact and how your body's recognized the drug. If we injected it into the blood, that drug would affect your whole body as opposed to just your lung. I like giving the analogy, it's like brushing your teeth and you scrub your whole body in toothpaste rather than just your teeth. It's just a wasted drug, and that drug's not going to do much else. So yeah, hopefully that answered that. And then what can be done to counter imperfect alignment of an inhaler? Great question. So there's a lot of electronics going into inhalers now. So looking at live feedback and connecting it to your phone based on angle. We can use physical guides such as ones that mount to your teeth or based on your nose. So they hook into your nose. But the bottom line, everyone is as different on the outside as they are on the inside. So it's really difficult to get a sort of one size fits all. So it comes down to the design of the product, but also the training that the GP and inhalation nurses give to the patients. So how many divisions of lung branching are you now modeling? So it's really only appropriate to use sort of 3D finite volume method to go down to generation maybe 6, 7, or 8. Past then, it's inappropriate just because of the number of branches in parallel. The scale just becomes incomputable. You couldn't get a mesh on that. So that's when we pass down to a 1D, something like a Markov chain model. So we need these two models, the 3D and the 1D, working together. And then finally, is the simulation workflow customized with the software? So we've developed this in StarCCM Plus. So we've developed that workflow there. So everything from the geometry prep to the physics to the post-processing was all done in StarCCM, software package I'd highly recommend. But I did work at Siemens before. What is the physical time for each simulation? I didn't mention that actually. Yeah, so these models run in about 2 hours on just a normal desktop computer, as opposed to like 10 days on a high performance computer cluster. So a lot more efficient. Who's funding this work? Pharma, NHS, other. So we primarily work with big pharma. They have the money to spend on this kind of R&D. But those insights and the products that they develop will be passed on to national healthcare systems like the NHS. And then elaboration on the 1D part of the model. So because it's inappropriate to model the flow regime, so turbulent, transitional, and laminar, and in one model, all in one 3D model. So by the time we get down to the 7th branch, there's 7 branches which have all split 7 times, is the maths right? So we have just a large number of branches in parallel, and that's just very difficult to first of all mesh, so to split up into finite volumes and then compute. So our compute time will go up, and as a lot of these airway paths are the same, they're going to be similar kind of diameters and lengths, we can just use a more statistical model of laminar particle deposition in pipe flows to predict where those drugs are going to deposit. I hope that makes sense. I can forward you to a paper if you're still confused, Alex.
I've got a question for you, Ben.
Yeah.
This is brilliant for the types of insights that are needed for the development of inhalation devices. But when you start looking at the regulatory pathway, when you start getting these devices verified, there's still a heavy reliance on physical testing. Based on the sorts of insights you gain from the simulation, are there particular changes you'd want to see in cascade impactors and other types of testing equipment to better assess these devices during that verification process?
Yeah, it's a really good question. So sadly, inhalers haven't really changed for the last sort of 20 years. There's been no significant improvements, maybe bar the soft mist inhaler. But really, because of the costs of developing new technology and because of something called the FDA 510K, it's a lot easier to develop an inhaler which is pretty much the same as something that already exists. It's a lot less paperwork. It costs a lot less money. And if the devices are selling, why would Big Pharma want to make new devices? But there are new devices coming, which we're aware of. And thankfully, the FDA is opening up to these kind of in silico evidence bases. So they use a weight of evidence approach. So we'll still need all the in vitro testing, the impact testing, but they will accept evidence from sufficiently validated simulation modeling. So you kind of need to do the testing to validate the model, to use the model to support in your evidence, which you're also doing to avoid doing the testing. So it's kind of a vicious cycle, but it can save a lot of work down the road.
Yeah, it reduces that burden of repeated testing for sure.
Yeah.
Great. We do have a final question from Alex. When do you predict the real world changes as a result of this work?
I think we're starting to see that now. So as I mentioned, there's sort of a next generation of MDIs with these new net zero global warming propellants in them. So some of these technologies are being used on those projects to make sure they're performing equivalently to the existing propellants. So really, we're starting to see those changes come to market now. But at Crux, we have the privilege of working on these next generation devices, and they are exciting, but they take a long time to develop because they're so novel. So that would be looking into the longer term future, sort of 5 to 10 years, where we can make an inhaler which will deliver the same amount of drug no matter who inhales it, and it will do a good job of producing a good aerosol which moves slow enough to reach the deep lung.
Nice. We still do have a few minutes, so we can keep on going through these questions. So we've got from Jack, will there be a CPT certification provided for the webinar? I see, that's actually one for me, I suppose, isn't it? There will be, if you go through the IET page itself, there's some more information about the CPT certification. But yeah, I have seen that there is a link for that as part of this event. We then do have another one that's then for you then, which is, will the new inhalers be more evolutionary or more evolution or more revolution? Oh, sorry, you're on mute.
Yeah, good question. Next 5 years, definitely evolution. But given some of the potential of the technologies that we're seeing, give in the 5 to 10 year range, we're hoping for revolution, which is really what we need. 20% drug to the lung is very, very poor, and we know we can do a lot better than that. Fantastic. I think everyone's out. It's bedtime.
Brilliant. Well, yeah, thank you again, Ben. That was a really, really interesting presentation.